slides. Those who have never been here in Hamburg, so Hamburg is the second largest city in Germany with about 2 million people, and you see here a nice picture from the harbor, because Hamburg is also the largest harbor city in Germany. All right, so today I will talk about a few things which are related to dealing with crystal structures, especially when we go from a raw PDB file to what we usually need in molecular design. So, and we have several topics we will cover along these lines. All right, so just to start with, everybody has seen PDB files before, and all people or scientists who work with this kind of data knows how difficult it is to really deal with this data. The tricky part is actually not just to parse all this information, but to go from here to the level of detail we need for molecular design. So here you see the CSAR front end from Biosolve-IT, and when you look at the part of the structure you see here in the center, you actually notice that there's much more information there than in the pure PDB file. So mostly the hydrogens are added, hydrogen bonds are shown, etc. Okay, so along these lines, we have a lot of things to do, and therefore we have 4 topics I will cover in this talk. So first of all, I will talk about this problem of hydrogens, so assigning hydrogens, dealing with tautomers and protonation states, which is also closely related to hydrogens. I will then go to some new things which you might haven't seen before, because this is really very new stuff, which has to do more with dealing with the amount of structures we have available today. So the first is an issue related to aligning binding sites, which come from different PDB files. And then in the third part, we will look at the process of easily collecting binding sites from the proteins, so that you can actually get an idea of the protein flexibility and can use all this structural information for modeling efforts. So in the fourth and last part, I will talk a little bit about water molecules, and we will not go into the direction of estimating energies or so. As I said, the focus here is on preparing information. So here we will look on how to select water molecules, and we developed some tools and analyzed water molecules in PDB files, and I will give you a short introduction into this. So that's the roadmap, and with this we can start.
Okay, before we go into more detail about what Matthias is going to say, we would like to know from you guys a little bit about what you do. So we have prepared 3 polls during the presentation, and we would like to know in this poll if you consider yourself either a computational chemist, a medicinal chemist, a biologist, crystallographer, or none of the above. I will start the poll now, and if you please enter your answer in one of these buttons, and I can see the votes coming in now. I'll give you a little more time. Yeah, I can already see that it's a neck and neck with between computational chemists and medicinal chemists. I'll give you a little more time, and I will close the poll in 5, 4, 3, 2, 1. Okay, so it seems like our audience is mostly computational and medicinal chemists. There are a few biologists and a few crystallographers, and some are even none of the above, or maybe didn't even want to share with us what they are. Okay, let's go back to the presentation, Matthias.
All right. Do you see the slide again?
Yes.
Okay, good. So So we will start with the first topic, hydrogens, tautomers, and protonation states. And so what's the problem actually? So we have about 88% of the PDB which are from X-ray crystallography, and this means that in nearly all of these cases you have no hydrogen coordinates, and this has several consequences. Most important, you don't know the positions of hydrogens, but the problem is usually even deeper, that you sometimes even don't know where the hydrogens are. So you don't know how to protonate amino acids or even the ligands. And sometimes, especially for ligand molecules, it's also the question which tautomeric forms the right one for the small molecule. This is very difficult to deduce from the pure 3D coordinates of the heavy atoms. And we have to think about what we can do here. This is just a reminder that this is actually a very important problem. It's just a comparison of two crystal structures, which were, these are kinase structures with small molecules. These are structures from publication from Loppe. And you basically see that the small molecules have only one small difference here. There's one acceptor replaced by a donor, or vice versa. And so this basically shows the influence of making a single hydrogen bond wrong, which you see here in the delta G values, which makes a delta G drop of more than 6 kilojoules per mole. So one wrong hydrogen actually has a drastic increase in the binding affinity prediction. So we have to be really careful and make these things correct. So it's not surprising that several people have looked at this problem before. So this goes more than 30 years ago. And so, for example, What If was one of the very early programs which has done these things. Many more are mentioned here. So this was the first version of Protoss we developed in 2009. And what you basically see is that all these tools take great care of the protein and also of the water molecules, but the situation differs substantially when it comes to the ligand molecules. And the reason is that for ligands, you have the whole organic chemistry you have to describe. So this is much more complicated than dealing with the typical 20 amino acids you have in the protein structures. So some of the tools started to consider, for example, tautomeric forms and protonation states, and these hooks here, they are all in parentheses, mostly because these tools have more or less collections of fixed tautomeric forms which could be replaced in the structure. Okay, so we tried to do this more exhaustively, and this resulted in the Protoss tool in the current version, 2013. And I will give you a rough introduction what it does and what the influence actually is. Here's just to give you an idea of what can go wrong. So if you look at the structures, you will immediately see the errors, but for the computational tools, it's sometimes not so simple. So here you see, for example, repulsive donor interactions, so two donors pointing directly to each other. What you see sometimes are hydrogen metal clashes, so a hydrogen pointing directly to a metal ion. Or you have even unsaturated donors and acceptors where actually the hydrogen is missing. You should either protonate the one or the other side. Okay, so this is just cases you recognize. Sometimes one might wonder why you see something like this and you say, yeah, well, I only have to remove the hydrogen here, but the situation is that you still have to create valid chemical structures. So this is sometimes not so easy. And this actually shows why it is so important to actually consider, for example, the tautomeric forms of the ligand molecules. Okay, to get an impression how important this problem is from purely numerical values, we have here the ligand expo from 1 year ago, which had about 17,500 small molecules. And if we apply our model for protonation and see how many situations we find where we can actually use different tautomeric forms, we see that in more than 80% we have ligands with altered alternative tautomers or protonation states. Even worse, we usually cannot solve the problem just by enumerating typical scenarios. This is something you see in this plot here to the right. If you just think you can enumerate the different fragments to consider the tautomeric forms, you see how many fragments you would need to describe the certain percentage of these molecules from the ligand export. So if you would have 200 fragments enumerated, you will come up to probably 80%. But you see that this is a very very small or very slight slope here. So you need thousands actually of fragments to describe this correctly. So this is not a good approach, and it would be better to have a more chemical-oriented approach which actually deals with all these structures. Many of these actually come from ring systems with so heterocycles, which have specific options or possibilities to protonate them. Okay, so we developed a tool to deal with this, which is called Protoss, and I will not go into so much detail related to the algorithm. It's here just to give you a rough impression how it works. We start with the PDB file. We identify all what we call variable groups, so groups where we might change either the protonation state, protonation protein, proton orientation, etc. We actually consider all possible states for these groups and score them. So we assume that the state is taken, and then we calculate a score for this group relative to the other ones. And this gives us a potential network where all these groups have different states. And in this, we have a specific algorithm to find the optimal network. So we don't need to do any kind of simulation at this point. This is done with the combinatorial algorithm to find this out. And from here, we can then actually assign the hydrogens and create the full structure. All right, to show you the influence of this, so we have lots of examples we could show in principle. So I've just picked one slide to give you an idea how important this is. So I show you here undesirable donor contacts in protein-ligand interfaces. So such a contact is undesirable if the contact between the heavy donor atoms is closer to a certain value, and then you have small angles here so that the donors actually point to each other. Yeah. You can have different feeling what's acceptable and what's not acceptable. This is here by these stars. So if you're very precise, you have very low tolerances here, and otherwise you might have higher values. So here in the bar, you basically see the consequences of this. Here you see the absolute frequency of donor-donor contacts, from which we can assume that they really do not occur in protein-ligand interfaces. And this goes from several hundred down to just a few which remain. And I was just point your attention to the two Protoss versions here, where you see this dramatic drop in the number of donor-donor contacts, which results mostly from the consideration of this tautomeric form. So the first version we had was not able to consider this. Now, when we are able to really handle tautomerism in the ligand molecules, we can really resolve all these problems. All right, again, just a few pictures so that this gets more real to you. Here is a quite difficult scenario where you can basically see the problems which occur. So you see here this this aromatic heterocycle. You see here in the first solution two mistakes. You have the donor-donor clash over here. You have an acceptor pairing over here where the hydrogen is missing. And this is clearly because for this ring system, the different tautomeric form was chosen. Yeah. So the second solution here was also not here it was also not able to find the correct tautomeric form. However, the tool corrected this by placing the hydrogen here on the protein side, which is obviously the suboptimal way, but even better than just missing the hydrogen bond. Yeah. If you're able to switch the protonations, the tautomeric form, you can go to the right structure over here where you have the correct hydrogen bonding network. So this is just one example of hundreds and thousands we looked at. One test case we have done is that we looked at the Astex diverse set, which is, according to literature, hand corrected, and we basically checked how close we come to the solution a modeler, an experienced modeler, actually created by hand. And you see here the number of errors, and again, if you compare Protoss with and without the tautomeric forms, you see that a lot of problems can be resolved by considering tautomers in this step, so that we believe that this is really an important issue here. So Protoss is usually running over the whole PDB, so we use this for protonating not only binding sites but also complete proteins, and we test by running over the whole PDB about more than these 100,000 structures which are available today. And you can also access PDB files with Protoss hydrogens. We are a web interface if you're interested to just look at a few of them. All right.
And this brings us to our next poll. If you are a medicinal chemist, of which I think 42% of you are, how much do you use software, obviously beyond Office, Mail, and Web? There are 4 answers, and I will launch the poll now. So please select one of the 4. Yeah, there seems to be a clear winner here. Very interesting. Almost 50% of you have voted. I'll give you some more time to choose an answer. And I'm going to close the poll in 5, 4, 3, 2, 1, close. So most of you guys use software all the time, which is not surprising. And also not surprising is that obviously there's still a lack for the perfect tool, I should say. And almost none of you like to draw on paper, although I have heard that medicinal chemists rather draw on paper than structures in the computer. Okay, thank you for this, and I will let Matthias continue with his presentation.
All right. Okay, so let's go on. The second topic I would like to talk about is about aligning binding sites. So this is a very new work which was published just, I think, 2 months ago now. And I will briefly show you what the problems are and how we address this. So actually, we are interested in creating ensembles of protein structures because we want to gather all this information which is available on proteins, and especially on protein flexibility when we do these kind of modeling endeavors. So you all know that finding or having the right protein structure might be essential. So here's just a first example, tyrosine kinase, and you all probably know these structures, which the DFG in and out conformation, which is a huge change in the structure of the protein, which make binding of the small molecule possible. Yeah, so looking at protein flexibility is usually done by either collecting examples from the literature, from the PDB then, or probably also by MD simulations, but if you do MD simulations, you don't have the experimental data in here. So therefore, collecting data from the PDB is a very nice approach. So in theory, it should be simple to do that because the proteins we are looking at are more or less highly similar. So the sequence identity should be extremely high because we are looking at the same protein. You might have 1 or 2 mutations. So a sequence alignment should do in general. And I will show you that this is actually not the case. And we start with a very simple example. So this is HIV protease, which is also a protein, a target which many of you might know. And if you have a look at HIV protease, you directly see that this is a homodimer. And when you work with the homodimer, you obviously have 2 possibilities while looking at the sequence, how you want to align them. The sequence don't tell you whether you want to do it in the one or the other direction, because these 2 sequences the protein consists of are identical. But if you look at the structure, you see differences because the symmetry is usually broken by ligands which bind to the protein. So here the red structure has different side chains orientations here and here. So it makes a difference whether you have the alignment of chain A to A and B to B, or A to B and B to A. Yeah, so this is something which can never be covered by sequence alignment tools. The situation can be even worse. So let's take another example. So here's uridine phosphorylase. And if you look at these structure here, you see that the binding region is typically between the interface of 2 of the domains over here. So a sequence alignment can always just consider one part of it, so one domain and not the other, and so that makes certainly a problem if you have binding sites which are located somehow between parts or between different sequences. This is an interesting example. You see annotations from the PDB by these letters here, because you might think at this moment, well, this is not really a problem because the chains have all their names, and if I know I see the interface between chain A and B, it will be always A and B. But this is not the case. So here are just 2 PDB codes with an inconsistent assignment. So in the green, so 1U, 1C, you have the interface between structures A and B, or sequences A and B. If you go to the blue structure, 1SJ9, A and B, which is the structure in orange over here, does not share an interface at all. Yeah, so looking at the annotations from the PDB works in many cases, but not in every case. Okay, so the usual answer at this point would be, okay, so we make structural alignment. So there are structural alignment tools, and that it doesn't fulfill all the needs we have for aligning binding sites. So here are 2 proteins. This is an ubiquitin complex. Here's a second part, which is not of importance for us. We are looking only at this region here, which is actually close to the binding site. And you see that there's a substantial change here, also in the secondary structure and the backbone fold. Yeah, if you do this with the structural alignment tool, the alignment tool will produce something like you basically see here, because it will try to fit the secondary structure elements on top of each other. But for considering the flexibility, you actually want to have the one-to-one mapping. So you need something which is closer to the regular sequence alignment, as we have seen it before. So our conclusion when we looked at all this was that we basically didn't found a tool which is able to really handle all the things you need for creating nice ensembles of active site structures. And if you look at large collections, so in docking papers, this occurs frequently, that the people need these kind of collections. You see that it is really typically a lot of handwork in creating these ensembles. So here are the requirements we see for doing these alignments. So we need an active site alignment only. We don't want to align the whole structure. We need a residue mapping at the end because we want to find out which amino acid changed the position. And we need this actually also for calculating things like RMSD or properties or descriptors. We have to handle the interfaces between protein chains because binding sites are often at these interfaces. We have to accept that there will be multiple matchings. So even in this structure, we have here 6 binding sites available, which all represent somehow the same binding site, which are just here at different positions in the crystal structure. And we have to be able to match the flexible regions, as we have seen here, where the binding site has a substantial rearrangement. So this brought us to the development of ASCONA, and ASCONA is an active site alignment tool, which is based on a fragmentation idea. It is highly sequence-based when it comes to looking where the fragments are in the structure, but it uses geometry to actually find out the right assembly of the pieces which form the active site. And what this means, I will show you on the next slide. So ASCONA has about 4 phases here. The query, so the protein binding site you start with, will be fragmented, so brought into smaller pieces. And so the pieces are connected to larger amino acid chains, which are in the binding site. So the blue ellipse here describes the binding site. From there on, we elongate these chains a little bit to have them larger, to make it easier to align them into the structure. Then there comes the fragment alignment step. There's something specific about it, but it's not so interesting. The more interesting part is then the fragment assembly part, because now for all these pieces, we find fragments in the protein structure, and we have to find out which fragments make one binding site. So actually, here we have a green one we can start with, and we have other green parts here which belong to the same binding site. And this is actually the part which is done by geometry methods. So we use a specific new developed a similarity function which considers distance and angle, so relative orientation of fragments separately, to actually find out which parts belong to each other so that we can create the binding sites. Okay, and from there on we can construct the alignment. And with this model, we actually are able to handle all these issues with binding sites between fragments, etc. Okay, so to see how this actually works, we started again with Astex data. Now we took the non-native data set, which is well collected data set of these ensembles. And from this data set, we took the portion where we have a clear PDB annotation. So a clear PDB annotation means that we have the same sequence IDs, the residue types are all identical, the insertion codes is correct, the chain name is correct, all this. This is the case for about 76% of the data that we have. So, and for these, we calculated the alignments with a method with ignoring this PDB annotation, of course. And then we compared it, whether we have the same annotation than the PDB has made for these structures. And you see that this is nearly always the case. So all the blue parts here are the cases where we have the same alignment calculated. All right, so the measure you see here is the distance, or the geometric distance between these structures, and I will show you just an example. So if you are here at the beginning, the structures are more or less identical. If you're here on the right side, you have a substantial change in the conformation of the molecule. Okay, so you probably have seen these 2 red spots here. This is actually one structure where we have a difference related to the PDB annotation. And here is the structure to give you an idea what happened. And this is exactly what we have seen with the hexameric structure before. So we see that the assignment of chains is not consistent. So while the interface in the green structure is between the domains A and C, the domains A and C do not share an interface in the blue structure. That's the reason why these binding sites are correctly aligned by ASCONA, because we do this by these structural fragments around the ligand, and where you are totally misleaded when you take the annotation in the PDB. So this was the one case we found, but you have to consider that we have only in half of the cases this PDB annotation. So in all other cases, we do not have this available, and we use this here only for validation. So here's the one case where, which I wanted to show you as an example for something which is strong deviation, to show you that these very difficult cases can be correctly aligned. So here, the structural alignment tool would most likely have problems. So the binding site is in this region here, and you see that's heat shock protein 90. You see the strong deviations you basically have between these structures. So with ASCONA, we are able to really get put the active site into the focus and calculate the alignments in a fully automated way. And in the next part, I will show you how we actually use this to collect larger ensembles of protein structures in a fully automated fashion.
Yeah, and this brings us to our next poll here. We would like to know your opinion about how much you think a good software tool could help you to innovate. Again, there's 4 answers. The first is not at all. You don't believe in software. Then you're somewhere in between. It depends. It's hard to find a good software though. Software can help, but most is done on paper. And the right kind of software would help us to be more efficient. So please give us your answers now. That's a pretty clear outcome here. I'll give you some more time to find an answer that fits best your opinion to this question, and I'm going to close the poll in 5, 4, 3, 2, 1 now. Okay, so most of you guys think, and that's a pretty clear answer, that the right kind of software would help us be more efficient, but it's probably hard to find this right kind of software because there's such a wealth of software out there. Okay, please go on, Matthias.
All right, so now we will go to the third part, which is strongly related to the one we have seen before, because now, since we are able to automatically align the binding sites, we can create a piece of software which automatically extracts binding sites from the PDB. And this is what we have done next, and this is completely unpublished. So you are actually the first to hear something about this. So this approach is called SIENA, and the idea is that we want to have a tool to instantaneously get all the binding sites related to the binding site of interest I have, which are available in the full set of PDB structures. So we start with one structure, and a click later we want to have all the structures available in the PDB. We want to have them aligned in the correct fashion. We want to have this no matter whether these are structures from or active sites from the same PDB file or from different PDB files, no matter whether they are homodimeric. So in this case, we want to see both orientations so that we have the most possible information we can get from the PDB out of this. Okay, you can imagine that with ASCONA we solved the key question for addressing this problem, and I will just give you briefly an idea of what we have to do now. So in order to make the approach fast, we construct a certain database, which is related to methods you probably know from sequence searching. So we take the PDB, we have a sliding window, we store short sequence patterns and put them into a database. Now, with this, we can start with the binding site of interest we have. We do a site fragmentation, so we put this into smaller pieces. And these small pieces can be used now to look up this, this database of small sequence patterns. And this gives us immediately a list of the PDB codes which have a high portion of the small sequence fragments we see in the binding site we are interested in. So these are our candidates, and with these candidates we can go into ASCONA. You've seen in the last part of the talk, so in the previous part of the talk, and then we can actually align the binding sites and check whether we truly have a correct alignment or a correct mapping of the binding sites. We then equipped SIENA with a lot of filtering options to so that you can really reduce the ensemble to the size you want to have. And more importantly, we have also methods to extract smaller ensembles, which might be necessary, for example, for docking calculations, where you can handle only a handful of different conformations. We also have a specific algorithm for the region superposition, so that you get the 3D alignment of the binding sites correctly and can have a look at it at the end. So this is actually something you in principle have already seen in all the pictures I've shown you in the ASCONA part. All right, so I will not try to go through all these steps in detail. Most of them are mostly trivial. It's basically the ASCONA part, which is the algorithmically challenging part. The rest is, I think, more or less obvious. And I just want to show you what we can do with such a method. So we again started with the Astex non-native dataset, and we thought about how you would actually try to extract the binding sites without having a tool like SIENA. So most likely you would take BLAST and make a sequence search and say, I will take the sequence to find out all the proteins I would like to align. So we started to compare what SIENA calculates differently than a regular BLAST search. So in this diagram, you see the 5 most frequent targets in the Astex non-native dataset. So we have most PDB structures. You see here, this goes up to several hundred, for example, for HIV protease. And here you see different settings for the tool. So the greens are the SIENA settings we have available, and the blue ones are the BLAST settings we have available. So here you can think about what you would do with BLAST. You would probably say, I want to have chain identity 100%, but this might cause problems. You will find only a few structures over here because you have minor changes in the amino acid sequence. So you can go down with this value, but you have to keep in mind that these values, the chain identity, are always reflecting the sequence identity of the whole protein, not of the binding site. So instead of this, SIENA is focused on the binding site. We can, of course, also make a global identity search and ask for 100% sequence identity. But we could also focus much stronger on the binding site. We can say we don't care about the whole identity. We want to have 100% sequence identity in the binding site. We can control the mutation rate, for example, in the binding site and allow a certain number of mutations, which might make sense. So you probably allow 1 or 2, but you don't want to have 10 or 15, yeah, something like that you can control. And again, you can do this for the binding site and not for the protein structure as a whole, because a mutation at this protein surface far away from the active site might not be that important. Okay, so you see the different number of PDB structures you extract, and you see if you basically go down with the values, you can basically find whatever you find with the sequence tool. But you have the possibility to really tailor the search to what you would need for calculations like virtual screening or the analysis of the protein structures. So let's look at one case in more detail. Here you basically have one test scenario here that's basically a hemoglobin structure and the things you basically find. So we look at BLAST first and we search for 100% chain identity, and then we basically see what we find when we use SIENA to basically show you that's not just a question of the numbers, it's also a question of the structures you retrieve. So if we go to a global mutation rate of 0%, we find structures like this. So we allow insertions which allow you to handle basically extensions of the structures like you see here, which is difficult to find out with the BLAST search where you can say I want to have 90%, but if you do that, you might have changes at different parts and not just elongations. So this is something you can handle by going from global identity to global mutation rate. We can go to a very high gap rate for the active site, and this is basically something to show you what happens if you do a BLAST search. So in a BLAST search, you can only search for one sequence, and you basically, in this scenario, you find structures like the red one, which overlays perfectly, but it doesn't contain the full binding site because it has only 2 of the 4 structures which are necessary. So, and 3 of them are forming the active site. Yeah. So sequence searches only allow you to search for one part of the structure, but not for a whole binding site, which might be composed of many structures. All right, so with the mutation rate control, you can actually choose to find structures where you have a mutation, like you have seen over here, yeah, where you and you can immediately control the amount of mutations you want to see in the structures.
All right, and this brings us to our last poll. We have one more question here for you. Do you regard ADME-Tox properties useful in a software, and would this be helpful for you? And there are again 4 answers. Of course, better check on those experiments. So this would be the early adopters here. Then for the skeptics, not without experimental validation. Third answer is sometimes it is good to have a second opinion, even if it is calculated. And then the fourth, for the ultra skeptics, I don't believe in ADME-Tox predictions. This is just guesswork. Okay, I'll launch the poll and give you some time to select the best answer. Okay, this seems to be a pretty even distribution, more or less anyway, except for one answer. Okay, I give you a little more time, and I'm going to close the poll in 5, 4, 3, 2, 1. Okay, so 40% said that it's good to have a second opinion. So that's obviously very advantageous for the software people here. And 34% actually really are strong believers in calculated properties of molecules. And there are also some healthy skeptics, and the ultra skeptics are only represented with 2% here. Thanks very much. This was for the polls, and I will let Matthias continue with his presentation.
Okay, so this was the summary of this was actually what we do for searching binding sites. So we have, as usual, a lot more examples and test cases and a lot of statistics. We basically calculated binding sites ensembles for every structure in the SC-PDB dataset. So a lot of things I'm unfortunately not able to show here. I can tell you that we basically haven't seen any cases where we would have to do any manual adjustment in the creation. And this is, for us, even especially if you think about modeling and docking, a huge step, because now we can easily create test scenarios for ensemble-based approaches like ensemble docking over the whole PDB, yeah, which is without any manual intervention. I didn't talk about the timing, so I can tell you we set up a web service in the moment. So we have timings which are measured over more or less the whole PDB, where we are able to construct ensembles in the time of 4 to 8 seconds. So it's just 4 to 8 seconds. It's more or less the time that you can easily use it interactively in a tool like Caesar, where you click on a button and you get all information from the PDB right at your fingertips. Okay, so from here I want to go on and we'll talk a little bit about water molecules. And as I said at the beginning, so water molecules are, of course, important. And in the last decade, I would like to say, a lot of tools have been developed to try to make energetic predictions about water molecules. You certainly know some of them. So we basically were interested at this point in a different question, a slightly different question. So because so much interpretation is done with water molecules, we wanted to know how much support we actually have when we talk about a water molecule. So here you see again a crystal structure. It's a thrombin structure and the water molecule over here. And this is a very important water molecule because with the change in the ligand molecule, here's a chlorine added, this water molecule disappears, and you can think about how this influences the binding affinity, which is a strong influence actually. So if you look at structures like this, and if you start interpreting the influence of water molecules, you should be really sure that the water molecule is there where you expect it to be. So it is important to actually compare the water positions with the electron density of the crystal structure. So here everything is fine. The water molecule is here. You see the cage here, which represents the electron density, which clearly shows that there's a water molecule at this point. Okay, so this is what we want to do, and I just want to show you already that this is not always that clear. So if you see a structure like this one here, if you wouldn't see the blue grids here, which show the electron density, you would see a lot of water molecules, and you would probably consider them all as more or less identical. But they are not. There's a water molecule like this one here, which is clearly resolved in the structure. So this is as a clear position, seems to be somehow fixed in the crystal structure. There are water molecules like this one over here, where you basically have no electron density support at all. So this structure is more or less modeled at this place because it maybe make nice hydrogen bonds or whatever, but certainly not because there was electron density. And even worse, you have situations like this one here, where you have a water molecule which is in electron density. However, if you look at the electron density, it is clear that this might be more an alternative location for the side chain than for the water molecule, because there's no clear difference between the water molecule and the boundary. So, and we want to know, and we wanted to automatically check whether water molecules are of one of these kinds. Yeah. Okay, that's the goal. And based on this, we can start to analyze really the water molecules and think about what are the structural characteristics of these stable water molecules, which are really there in the crystal structure. Yeah. All right. So this is actually what we want to do. And just to remind you, these water molecules are coming usually from modelers, but also from software tools which basically place water molecules, so that it might be that they are false positives. Okay, this one I have shown you already. This is one with high electron density. Here is an example without electron density. So there are, of course, several measures which allow you to measure the support for different atoms in the electron density. However, all these measures only consider the whole structure at once. Yeah, but we want to know now the single water molecule. Is there support for it or not? And for this problem, we developed a measure to do this. So we work with the electron density map, which is then resolved on the grid structure you can see here. And we make the calculation with the function we called EDIA. And due to lack of time, I will not go to the formula, but this is was published earlier this year. You can easily look at the details in this publication. More importantly, I basically want to show you what we have done with this. So we have a collection of 5,500 protein structures from PDB with certain criteria, and so this should be high resolution structures. And for us, it was important to have the electron densities available. And if you look at these structures, you find 2.3 million water molecules. Yeah, and now we can automatically check with this EDIA score for each of these 2.3 million water molecules whether there's experimental support and how strong this experimental support actually is. Yeah, and so these are the EDIA values you can see here, and here are the number of water molecules which you see in this set of 2.3 million values. So to give you an impression what these numbers actually mean, they are not normalized as you see here. I have just a few examples. So this is an EDIA value of 0. Yeah, you basically see a structure like this, a water molecule with no electron density support. This is something you see here. And I hope you immediately recognize how high this bar actually is. Yeah, so there are a lot of water molecules there where you have no electron density support. This is a low EDIA value of 0.21. This is a value of 0.25. So as you can see, so here it is still a very small signal only. Here you have a larger signal, and this is the borderline we set for saying that the water is really there. So we use an EDIA value usually of 0.24 to say that that the water molecule is really there. And then when the EDIA value goes higher, we have situations like this. So here we have a value of 0.8 roughly, where you have more electrons and a higher electron density. And the situation, if you go higher than that, is that you usually have the situation that you modeled water molecules into structural pieces which are not water, which could be either parts of the protein. Often these are metal ions, which are then used and waters are placed in there. Okay, now you can do a lot of analysis with all this data. So we looked at hydrogen bond characteristics of waters in these PDB structures. You can count the number of hydrogen bonds you see. You see that if you have electron density support, you have a higher number of hydrogen bonds. We have a classification here for surface complex water molecules, or covered water molecules, and these are in protein-ligand interfaces and protein-protein interfaces. Again, a lot of statistics which you can do, and a lot of things one can do when you look at water molecules with this additional information. Just one thing we were certainly interested in are so-called hydrophobic bubbles. So are there water molecules with a clear crystallographic resolution which do no hydrogen bonding at all? And with EDIA we can answer this question, and we see that about 0.5% of all captured water molecules. So a captured water molecule is a water molecule which has protein or all around. Yes, so in a situation like this here. So no other water molecules or ligand. So every 200ths of these water molecules is can be considered as a hydrophobic bubble. So the water molecule sits there, no contacts to the protein. So no contacts, no hydrogen bonds to the protein, and so you have now 1,400 water molecules, and there's certainly a lot of information you can learn something from. Yeah, by looking at these water molecules, understanding why they are there, what's the biological relevance? Why are these water molecules there? They must be highly suboptimal. They can be used for optimizing ligands if they are close to binding sites, etc. Okay, so we also have looked at the other cases. So we looked at 4 hydrogen bonds, and say water molecules with 4 hydrogen bonds, there must be a clear signal that they are really in the structure. And here you see situations where this is not the case. So low EDIA values with these water molecules, situation like this here, yeah, where we also don't know why the water molecule might not be here at all, yeah, because it can form 4 nearly perfect hydrogen bonds at these places. Sometimes we see water molecules in these water clusters, yeah, and these are highly resolved at some point. Some of them are missing, which might be a result of the flexibility we have in this water cluster over here. EDIA values which are very high are also interesting because you can find situations where there might be errors or at least say strange things in the structure. So an EDIA value which is higher than 3.3 is most likely not a water molecule. We found more than 1,200 with EDIA values of that size, and we took our time and looked at 10% of them, yeah, so 120 cases, and then we were not able to do any more. And we more or less exclusively see cases, well, in 75% we see cases where we have the opinion that this is a clear misinterpretation of electron density. So here you basically see a water molecule, and we have superimposed an octahedron just to show you that most likely this is a metal ion here from the interactions this is able to form. Or situations like this where you have a water molecule, and this is very likely an alternate location for the sidechain over here. Okay, more on waters you can find in this publication here. As always, time is much too short to say all the details of such a study. And this closes the 4th part of my presentation. And I come to my conclusions. I hope you all agree that protein structures are a rich source for modeling, and that we should use them as much as we can. Crystallography is a great tool for which allows us to all these things, but there's a certain gap between what comes out of a crystallographic experiment typically and what we need for all this detailed atomistic modeling we are doing. And so it's important to close this gap and to do this very precisely. Yeah, and so today I presented 4 tools for you. Protoss for hydrogen bonding and for hydrogen placement, for tautomerism, doing all these things right. ASCONA and SIENA, which help you to collect all this information, work with flexibility, create ensembles of structures and learn from them. And the EDIA value, which helps us to look at the water molecules, which are so important in many situations of drug design, but where we really have to do a critical review whether they are available in the structure or not. All right, so a short picture of my group in the currently, and I will not go through all these names, but I want to show you the 2 who have done most of what you've seen today. So the EDIA value is done by Eva. She's standing here in the back, and here in the front, this is Stefan Bietz. So he has developed all the things we have seen around protein structure, starting with Protoss, and later on also the ASCONA and the SIENA approach. All right, and with this, I would also thank all our partners, companies we work together, funding agencies, etc., and certainly Biosolve IT, because you might have noticed that we do a lot of software development in the group, and doing this means that you really need also the support for professional software development, which goes just beyond what you usually can do in an academic group. And the collaboration with Biosolve IT really makes this kind of method development possible. All right, with this, I would like to stop. There's a lot of information you can get. Here you see the web address from my university group here. You have a list of all the tools. You find all the papers we published over the years here, and a lot of additional information. You also find information about software availability and web services we do, and that's the address of Biosolve IT. They also host a lot of the software we have done in the past. Okay, and with this, I would like to stop and give the word back to Carsten.
Yeah, thanks very much, Matthias, for this excellent talk. And I would like to knit on here, so to say, right away, and showing you where you can find these developments that we do together with our prime academic partner. We have a website here, biosolveit.de, TechTrans, for technical transfer. And this is where we host some of the most exciting developments, and the most useful ones actually make it into the software, such as Protoss, which is an integral part now of CSAR. We calculate in really a fraction of a second the hydrogen bond network between a ligand and a protein, and within the protein, and this with very much accuracy, as you've seen with on one of the slides of Matthias, that also the tautomeric states of the ligands are taken into account. So this is an integral part of CSAR now, and those that have not found their way into the software yet are and will be available here on this tech transfer site. So that's for your information. Academics can download these tools for free as soon as they appear here. And I'm sure some, if not all of these that Matthias today introduced to us will be available at some point on this website here. So now, this here is the TechTrans website, and this is where you find these exciting developments. Again, the URL is www.biosolveit.de/techtrans. And there also have been quite a few questions along the way, and I hope you don't mind, Matthias, to answer some of them.
Sure.
Okay, so we have the first question here. How is ASCONA different from generally used servers like CAST-P, DogSite Scorer, etc. for active site predictions?
Okay, so ASCONA doesn't do an active site prediction. So DocSite Scorer, for example, or other programs, they use one protein structure to find out where the active site in this one structure is. This is a very important first step. If you start with one structure, ASCONA actually addresses a different problem. ASCONA solves the problem that if you have two proteins, and for one protein you know the active site, you want to have the second protein aligned to the first one in a way that you have a best match of the amino acids in the active site to have them on top of each other. So it's just a different kind of problem which is addressed here. So actually they fit well to each other. So a tool like DocSite Scorer or CAST-P gives you one active site, and with this you can use ASCONA to align other similar active sites on top of this.
Okay. And another question is a little more practical. How commercial software, paid software, is different from these freeware tools, and the listener would like to know if it's as reliable to use as commercial software packages.
Yeah, that's a more political thing to answer. I think we are all scientists. It doesn't matter where you are, whether you're in academic or in a company. Companies sometimes have much more resources to develop things. I think it comes back to the research group and how they actually develop and design software. So I think the borderline cannot be whether it's a freeware tool from academic or a commercial tool. I think you find good and bad on both sides. Well, yeah, that's I think all I can say about this. Uh, the one thing I would like to add to this is that uh, from the academic side, I think it's sometimes easier to get a realistic picture of what you can really achieve with a software tool.
Okay, then another one asked if it's recommended to remove the water molecules, because in other tools such as Autodoc, they actually recommend to remove the water molecules. Can you say anything with respect to that?
So yeah, if you're in the docking scenario, it depends a little bit. So if you do not do any analysis of the water molecules, it's certainly good to remove the water molecules, especially in redocking scenarios when you just want to see whether the docking do the right thing. If the water molecules remain in the structure, you really bias the calculation towards the ligand molecule which is in the structure. But in a docking calculation, you usually want to dock other molecules in the protein structure. And in this situation, water molecules will change their position. They will be, of course, removed during the binding process. And having the water molecules there will predict these binding modes correctly. So in general, yes, it's good to remove all the water molecules. But then, on the other hand, if you then look at these EDIA values, there are some water molecules which are really well resolved in the crystal structure. If you take multiple structures, like you can do now with the ASCONA and SIENA approach, you can even check for water molecules in how many structures they are available. And then you can get the feeling about the importance of a water molecule for binding. And then it sometimes makes sense to selectively keep water molecules in these experiments.
Okay. Another listener asked how Protoss is used. Is it applied retrospectively to already solved crystal structures?
So when we yes, so we actually start with the crystal structure and then we add the hydrogens. I'm not 100% sure, but I assume that the question goes into the direction how we really evaluate Protoss, which is really a big problem because we have nearly no structures available with hydrogen coordinates on it, so that you have something like a ground truth where you can say, if I make this prediction, it's correct. This is why I've shown to you these examples where we argue about the correctness of the structure, where we believe that this is the right hydrogen bonding network you basically see. Yeah. So yes, we apply it retrospectively, but in general, the method is fast enough. As Carsten said, it's part of CSAR. So we can do these calculations in fractions of a second. So whenever you place a ligand molecule in the binding site, we can use Protoss to reassign hydrogens and reorient hydrogens. And I think this is a very important thing, so that you don't do it just once when you prepare the protein structure, but you are able to do this on the fly whenever you change something in the structure, or when you change the position of a ligand molecule, or you place a different molecule into the binding sites.
Mm-hmm. Okay, well, I think with respect to time, we should stop at this moment. Unfortunately, we don't have time to go through all the questions. If you still have a question that you think is very important to you, you may send an email to us at Biosolve IT, and we will make sure that the question gets forwarded to Matthias, or send it to Matthias directly. Yeah, at this point, I'd like to thank Matthias again for this excellent overview about the most exciting developments in his group. And I would like to thank all the participants today for taking the time to listen to our contribution. Two more hints that I have to say is that we will make a license to CSAR available to everyone who participated today. The license will be sent out in the next few days, so please be a little patient with us until the licenses are sent out. And this webinar has been recorded as well, and a recording will be available on the website on the webinar website, and you can listen to the webinar again and maybe enjoy the slides again if you missed something during the presentation. So at this point, I'd like to close, and thanks again to everyone to make this webinar a success.